You're going to get the pain of me addressing you directly now though, as well. So thank you for those on Zoom and for those in class. We are going to finish off our bioconductor sessions. So just to remind everyone where we are, rockefelleruniversity.github.io is our page, and there I've been trying to keep up with the videos. I think I've got everything we've done so far up until...
Welcome to the YouTube channel.
Up until the last bioconductor, which I think was alignment. So now we should know how to align our data. And we're moving on to the next part here, where we moved into bioconductor. We've run through an intro. We looked a little bit about how we can handle sequence data and genomic intervals. On top of genomic intervals, we thought a little bit about how then we can make genomic scores. So how we can turn or make use of genomic intervals with an actual value telling you you have this much signal, this much conservation at a particular region. So genomic scores we often see as bigwigs. They're graphs of signal from, say, your RNA sequence chips. We then had a little look at genomic features, which is how we handle genome models. And we saw that we have genome models available both inside Bioconductor packages as the TXDB database packages, but also the org-DB packages. We have annotation on gene to gene symbol to gene ontology. All this is held within Bioconductor. We also saw that we could use our own GTF GFF. So the issue being your lab may not be as committed to the bioconductor annotation as I am, but you can still import your own GTF, your own GFF, and make use of the really fantastic tools available within genomic features packages. So in our last session, we started to really go back to basics again, and we picked up FASTQ, FASTQ being what you will get off the Genome Center faster with qualities. And we saw a little bit how we can import this, look at some really important features. Basically bias across cycles for read, for base content. So do we have more G's or C's across cycles? Does it change? In total, do we have more G's or C's in our reads than we'd expect? And then we looked about how we can potentially do a bit of filtering, a bit of manipulation of our FASTQ, and then write that back out to another FASTQ. We also very briefly introduced you to rFASTP, which is our own software, and that's written in C++, but you can access it through R. In our last session, we introduced ourselves to aligners. We learned a little bit about, briefly, we talked about indexing, but more importantly, we learned how to turn our FASTQ into aligned data, either using a standard aligner or one which is splice-aware. Splice-aware being particularly important for RNA-seq, ribo-seq, anything where we're really not sequencing the DNA, the genome, but we're sequencing cDNA transcripts. So in today's session, we're going to have I think the first session is quite short. Second session is a little bit more intense. But in our first session, given that we've now taken our FASTQ, aligned it to the genome, we can start to think about how we can handle this alignment. So align data in Bioconductor, and we're really just going to talk about this BAM files. In reality, I think most of you don't need to necessarily interact with a BAM file at this level. You will pass your aligned data to some of these soft programs, functions we'll see in our second session, or you'll pass it to external software. What is nice about being able to look and align data is, just as we can review things in IGV, being able to look at your reads, look at the content of them is really important for quality control. And as soon as you start doing something custom, you want to be able to interact with the reads, right, the actual alignments. So aligned data in BAM format, let's remind us what this looks like. Yeah, say BAM format, but we wouldn't be able to read this if this was truly BAM format. BAM is binary alignment map. It's readable by a computer. we are actually looking at SAM here, and that's sequence alignment map. So what is in a SAM file? We have, do I actually go through it in detail? Yes. So the first thing in a SAM file is a SAM header. Okay, so this is our aligned reads, but before any of the actual SAM, or before we see any of this information in our file, this being one line, this being another line, this being another line, we have a series of lines at the beginning of our file which tell us a little bit about what's coming up in the file. Okay, so this is important because BAM files are potentially huge, gigabytes once compressed. And we don't want to necessarily read through the whole file to understand what's going on. So if we've generated it, a SAM or a BAM file, we should have this header, which tells us a little bit more information. And, we can see here the first line tells us the version number of the SAM file. It tells us it's coordinate sorted, which is important because that's going to save us time. We don't need to sort it. All the reads are organized by their alignment position on a genome. And all the chromosomes, so reads are then grouped by chromosome as well as sorted by coordinate. We can actually see the order of the chromosomes here as well. Okay, I'm not sure how relevant it is, but I can tell you that, one of the tricks that SAM files, BAM files use to save space is they don't bother writing out the chromosome name in the file. They actually just say, we are the second or the first in the header. We are the second in the header. So, it would say, I am the first in the header. It doesn't need to store chromosome 10. It just looks it up as it starts to write it out. Another important information here is ln. This is length. So this tells us the length of the chromosomes. Oh, from here to here. Sure, I'm not doing something awful, chat, but, which is useful again, because actually that tells you a little bit about what genome you are. So if you're not sure, someone tells you this is aligned to HG38 or a different genome build, you don't know which version of the human genome build is on, you can actually check these lengths and say, well, for HG19, they had this longer chromosome. I know I'm on HG19. In the actual body of the BAM file, we have all the information on the original read. So remembering FASTQ was 4 lines per entry, and we had the FASTQ read ID, which contained the machine, which is really useful for debugging. You know what machine it was, but also the position on the machine, on the flow cell on the machine. We have the sequence here, and we have a quality scores corresponding to every base pair in the sequence. Pardon me? It's telling us how good we did, how well sequenced that particular base is. So if we had a low quality score, and I think we checked this, we looked at hash was a low quality score, the corresponding read is probably not very confident. We're not really sure that that C is a C at that position. So say here where we have a hash, we're not sure how. We don't really care about that now, right? We've already aligned our data. Hopefully we've trimmed out poor quality reads, but the aligner will take care of low quality. It does this just by either ignoring the bases over it, down weighting them in the alignment, or sometimes it will just trim them out as we go. So it won't use that part of the read for alignment. We'll use the rest. But we use it to tell us how good we've done and how the quality of our sequence. Other really important things, or the most important thing about a BAM file, is its chromosome, its map to, the position on that chromosome, then the type of mapping we have. So chromosome 2 at this position. I'm not sure if I'm going to go into it. I'll go into it a bit in the talk, but here we have 100M. This is our cigar string. So it doesn't say maps from here to somewhere else, right? It tells us it starts mapping there, and then from that mapping, we have 100 matches to the genome in a row. Okay, start there, move along the genome, 100 matches. This one's a little different. I don't highlight it here. I think we've looked at it before. This starts here, and it has 28 matches. Then it's missing. There's a deletion in our read compared to the genome. And then we have 72 matches. So we know the read's 100 base pairs long. somewhere we don't have a base which was in the genome in our read. So we can call that a deletion in our site. We can handle aligned data through effectively an extension of genomic ranges called genomic alignments. There's a lot of packages called genomic something. Genomic alignments is built on other packages we've already encountered. So our SAM tools we've discussed a little bit. This is just the R wrapper for SAM tools. And SAM tools is one of the fundamental tools in handling high throughput sequencing data. Genomic ranges we saw covers range information. We know we're going to need range information because we've got mapping here. We've got the concept of how long the map was. Short read that was how we handled FASTQ. And as we saw here, we have all the information for a FASTQ within our SAM file. So we're going to use short read 2 to handle that. BS genome allows us to handle sequence information, and our track layer was our IO package. So really, to use genomic alignments, it's good that we've covered some of these other packages like genomic ranges, short read, BS genome, our SAM tools, our track. So we saw in our previous session how to align data to the genome. Shouldn't be that complex. Really, we're going to have to make a choice between what is our aligner. I'm sure people in your lab will have a choice. Our HiSat2, our subread are popular aligners, which we use. The big difference being, are you going to map in a splice-aware manner? So are you going to take into account junctions, or are you going to map like normal DNA? You're not going to use a splice-aware. So we have some data available to us for this session. So what I've done is I've taken the IGV example data, which they have hidden somewhere in their file systems. I've extracted that, I've realigned it, and I've placed it into a BAM file aligned using subread. So first off, we'll start just being able to get this BAM file in a decent format. We covered a bit about the RSAM tool session in our last sections to show us how to process, just perform the next steps on our SAM file, BAM file to get it into something usable. So RSAM tools is the basis for many packages. Anything dealing with alignment is probably going to use the RSAM tools. So the first thing we're going to do is load this, and we know how to install any package hopefully already. We can just search with Google, takes us here, and we can Control F, paste that into our session, our R session, and that will install the package. Hopefully we've already installed it because we've already used it in our previous sessions. So we can do library RSAM tools, load that library and all the functionalities available to us. We've already seen that one of the most useful things from SAM tools is sort BAM. Hey, great, thanks. Yeah, we're in the middle of it right now. Thanks. What's that? 12:30. Thanks. So we saw that we can do organize and get our BAM files sorted by chromosome name and then coordinate using this sort BAM function. So actually what it's doing is it's going to look at our chromosome order in the header of our SAM file, and then it's going to try and organize them by chromosome and then sort them by coordinate. So this again gives us our nice, without any other definitions, we just need to give it our BAM file. Remember, we give it our output prefix. This is annoying, but it's going to add .bam to whatever we put here. What is nice is sort BAM returns the name of the object it creates, the file it creates. So we don't need to worry about this too much. We get back the name of the file it's created, sorter_liver.bam. Some external programs will require that reads be sorted by read name. Okay, so get back a bit. So we could check, obviously, our sort order. It says coordinate here. Sometimes this is going to need to say query name, right? And that means it's going to be sorted not by their position of alignment, but it's going to try and place reads next to each other based on this. This is the read name, right? So if we have paired data, actually you'll have, this will be twice in your BAM file. You'll have one for read 1 and one for read 2. So you can imagine it would be easier for programs if they want to deal with paired data to have the pairs right next to each other, because it can just read the information of pair 1, pair 2. It knows where the limits of the fragment, where that sequencing has come from, and then it can do its analysis without having to hold reads in memory while it waits for its pair to pop up in the BAM. Okay. Good examples of programs which require that is bed tools. I'm not sure if anyone uses bed tools. It really works a lot better if you use query name sorted files. So we can do that very simply just by adding by QName, setting that to be true. And then it's not going to bother about coordinates. It's going to find read names, place them next to each other, and write them to file. Okay. So you can still do that, even though we're not going to use that much inside R, because what R does is read this all into memory. So we'll have all the read names available to us. When you're doing a program like bed tools, which likes to stream it, sorting by query name is really important. So now practically, if you are going to use R samples like we use R samples, you're not going to be able to run and sort your proper RNA-seq BAM files on your computer in a memory-friendly way. If you do, maybe you'll have a nice laptop like me, or you're working on the HPC, and that's possible. But if you are limited in any way by RAM, the way sorting BAM files is actually works is actually very easily split. So what we can do is actually set the maximum amount of memory that we want to use to sort our BAM file. And what it will do is create smaller sorted BAM files, and it will place these on your computer. So, we're going to reduce the memory used by our samples here and sort BAM, but it's going to produce more temporary files. So it's going to sort them in smaller chunks, and then it's going to merge the sorted BAM files. Okay. We can control this here, and this is a really extreme case, don't do this. So here I'm going to sort this tiny little BAM file, and I'll just use 1 megabyte of memory. For you might want to set this to 1 gigabyte. You may decide you're on the high performance computing cluster, you're on some supercomputer, and then you want to set memory to be way high, because you can just have it all in your memory. You don't need to worry about temporary files. So I'm not sure what the default for sort BAM is. I think it's 8 gigs. Which seems a bit optimistic that we would have 8 gigs on our computer. But we can look. Oh, it's very small, 520 megabytes. So clearly, this is probably set by older laptops. We could raise that to, a few gigabytes. That will, lowering this will mean you use less memory, but it goes slower and you create more temporary files. Raising this and having the available memory will mean it will go a lot faster. For anyone who actually runs our SAMtools for real, there is a parallel version of SAMtools. This just hasn't been put into our SAMtools yet, so we can't use that. Once we have our coordinate sorted file, it has to be coordinate sorted for this, we can then index these files. And the really important thing about indexing a BAM file, is that we can do random access. So really, this is going to allow us memory-friendly operations later on. You may be interested in just looking at one gene in IGV. You may just be looking at one gene to study the repeat or something. You just want to look at the reads over that region. Maybe look how many repeats you can find in the read. Indexing effectively creates this binary file, and then we can read the binary file to find out where we need to look in the BAM file. So lots of programs require these BY files, and they won't even let you accept. They won't accept a BAM file without it. We can create these really easily just by running index BAM, and that calls SAMtools, and that runs our indexing and returns to us the name of the index. It's actually a named vector, and the name is the file it's an indexing of. So this is one way of having it. You may find that sometimes you just get sorted liver.by from other external programs. but they will always end in a .bi, binary alignment index. So we saw in our last session, we can use this quick BAM flag summary. Okay, I'm not sure if anyone actually tried running that. So quick BAM flag summary is slow. Okay, just by the name, quick BAM, it's actually pretty slow, and it's going to go through your reads, one read at a time. And it's going to evaluate them, read the flags, and then give you the result. Okay, we get useful information, but most of the information I really want to get from this is how many reads I actually have in my file. Okay, how many were mapped. All right, take the nick out of it here. So yeah, despite the name, it really is not that fast, and it's just because it has to do a lot of work. To get a really instantaneous summary of how many reads are actually mapped within your BAM file, we don't need to look at the BAM file at all. We can look at the binary index file, the BAI file. Okay, so we can use this function IDX stats, so it's index stats. And we just need to give it the path of our sorted indexed BAM file, so it needs to have an index clearly. And in this case, I've just aligned it to chromosome 12. Okay, because we just have these chromosome 12. It's this short little liver map, body map example. So we can see it gives us seq names, it gives us the chromosome name, the seq length, and it gives us the number of mapped and unmapped reads. Okay. Clearly, you can't be both unmapped and on chromosome 12, but at the end here we have star, and it tells us we have 107 unmapped reads. So our SAM tools is the program which allows us to call effectively SAM tools. So our SAM tools is a wrapper for the SAM tools software. I'm not sure if I showed you SAM tools. That's SAM tools organization. That's what I wanted. Here we go. So this is SAM tools. It's a downloadable piece of software. I believe you can download it for most operating systems. And effectively, this just works on the command line. So that's really under the hood what our SAM tools is calling. Just allows us to operate on things like sorting, indexing our files. Really, if we want to import BAM files into Bioconductor, we're going to use this genomic alignments package. So we can install this like we do with all packages. We just search for it in Google, and I come to the page, and then we can copy this little section here, paste that into RStudio, and we'll have the library available to us. To actually load the library, we do library genomic alignments. There's nothing in chat. Okay. So we saw earlier on, I think I've already described this a bit, files, and the BAM files contain header information, and this header information contains a list of chromosomes used and their length, as well as the current state of the BAM sorting. So is it unsorted, sorted by coordinate, or by query name? Really usefully, additional information on the programs used in the processing of this BAM file are also present in the BAM header. So if you get a BAM file from someone else, or you download it from ENCODE, you can actually look in the header, and if they've been good, like ENCODE has, or like we are as a core, you will see that it has program information, how that program was run, what version was run. So SAM files are reproducible in that way. They tell us actually how they were created. So the first thing, to get access to this header, we can just do scan BAM header. We give it the path to our sorted index BAM file. It doesn't actually need to be indexed, I think, in this case, because we're just looking at the header. It's always at the top of the file. If we do this, we get back a list. I don't think it's a clever object. It is just a list. And in there, we have one element long, and that's actually the name of the BAM file we're looking at. And inside that list, so we have a list within a list, we have the targets, which are going to be the chromosome names and lengths. And we have the text, which is going to contain, as you can kind of see here, some program information. So we can use our hopefully very comfortable accessors now of list-like accessors like dollar or double square brackets to access these objects. And I think we've talked about this before. Most of the time, if you don't understand an object, it will define what you can do with it when you just type it in. Most of them act like lists. Dollar, a double square bracket will allow you to interact with it, extract elements from it. So we can use the dollar to access the elements in our list. And from there, we'll get either the targets. So we could do my header, my header dollar sorted bam dollar targets, or my header dollar sorted bam dollar text. And that will extract these various information. If we look at this header, maybe I'll just quickly read in this header. I think I did it earlier on. Oh, I've got the header. So just to give you a better idea. So my header contains a lot of information here, but the first level list is this sorted liver.bam, dollar sorted liver.bam. Then I have two lists underneath that, one called targets and one called text. If we look at the targets, it's just a named vector of the length of every chromosome. Here, I've only got chromosome 12, right? Because we just took the reads from encode or body map, sorry. But I have, similar to what we had in the actual header itself, where we have this information here. This could be considered the targets. It gives me the name, and it gives me the actual length. Really usefully, we have the text, and we can see in this, we have the HD tag. So I can extract this HD tag here just using square brackets because I'm extracting the list itself. But this tells me the version number of the SAM tools we're using here, just version 1. And importantly, it tells me it's coordinate sorted. So that would mean I wouldn't bother sorting it. I would save myself half an hour, an hour. If I'm doing 100 files, that's going to save me a lot of time. And I will just read this and say, nope. We can also check for our name sorted BAM file. So in this case, if I got a BAM file, I did my scan BAM header, gave it the path to the BAM file, and I check dollar name of the BAM file, dollar text HD. Here I can see its query name. So previously we will have unsorted coordinate, but now we have query name. So I probably would have to go and resort, okay, unless I'm using a tool which wants query name sorted. Again, it allows me to save time, not read the whole BAM file, not sort the whole file if I don't need to. We can see the program information by reviewing inside the text element. So again, this text element, we can go get this PG tag. Okay. And here it's depending on the program you use, which is why I like HiSat, it's why I like subread. They put different types of information in the BAM file. Because this SAM file / BAM file is created by those aligners. So it has, every aligner has a slightly different setup for what it wants to put in this program tag. But most of them follow this convention. We have the ID. So what is the ID of the program? We actually have a program name, which is again the same. We have version number, our subread 2.1.0. And then we have this final line, which is actually the command line command. And although we're calling this from inside R, subread is just calling another subread C++ program. We can actually go and really look at this in detail. Text, what was it? Dollar program for. I'm going to use this command cat here, because cat just, it's like the Linux version of cat. It just writes your, it's going to print this, but it's going to take into account all this little bit of escape characters we have. So things like line end characters is going to evaluate for us. Oh, sorry. So I run that. We can see we have the full command for subread. So we have subjunct, sorry, we have the reference, or the reads to align, sorry, that pass that file. We have the output. We have the index, which I called chromosome 12. And then we have all these parameters we didn't even list. Right? These are all the parameters which subjunct takes as default. So this is incredibly reproducible. And given that you had these files, someone else could pick this up, run your data again, which is great, because most of the time it will be you trying to rerun your data. And that in combination with the version number makes this very reproducible, just the BAM file itself. Sorry, lost my cursor. Okay. Now we have an idea how our BAM file was constructed, and we want to review some of the data in our BAM file. So we can use a very simple function, read GA alignments. Read GA alignments, and that will give us back a new class we haven't come across about yet, a GA alignments object from the genomic alignment. If we just type this in and look at the first two reads, we can already kind of get a sense of what this looks like. It looks a lot like a GRanges with more information. We have seq names, strand. Again, we have these bloody RLEs. We have width, except here it's called Q width. And then we have a second width here. We're going to explain what that is as we come along. We have the start and the end of the alignment. And then we have the number of junctions in the alignment. So chromosome, strand, the cigar string, how it's aligned. So this is aligned 31 matches and then 19 soft clip. So I was telling about how aligners may clip off the end of reads if they believe they're low quality or don't map. This is soft clip. The Q width being the width of the original read, the start and end of the alignment, and then the width being the width of the actual mapping to the gene. So even though the read was only 50, the read was 50, only 31 bases of it actually mapped to the genome because the last 19 were soft clipped off. The other way around here, where this one's mapped to the positive, 20 was soft clipped off at the beginning and 30 mapped here. And we can see that reflected again in the width here. So as I say, they're very similar to GRanges objects. We have additionally this Q width, whereas width is the actual alignment on the genome, which is really what we're interested in. Because we know how long the actual read is, and we know the cigar, we can also extract Q width. Apart from that, it looks very much like a GRanges. It also contains the cigar. Okay, so we already know a little bit about cigar, 75M, 75 matches in a row. We have the concept of insertions, deletions. We have the concept of junctions. If we look here, we can actually see an additional column called njunc, which will be very useful later on. This tells you how many junctions this particular read spans. So if a read maps across splice junctions, we can extract those reads directly by just filtering down on this njunc column. So again, GA alignments inherits most of its functionality from GRanges. We do not need to learn much more. We can do seq names, my reads, and we'll get the chromosome names as we did before, like a factor RLE. We can do seq levels, remember, if we want to change our seq names. We can do starts, just like we did with the GRanges, and it will extract our start and end, sorry, our start positions. We could do end, like we did with GRanges. We can do mcols. Additional accessors, which we didn't get with GRanges, are cigar and njunc. And the actual reason we need these as accessors is because, as we'll see later, R is calculating the cigar and the njunc de novo. It's not read them from the BAM file. It's actually going to calculate them from the alignment. So we have cigar, and if I just give it our GA alignments function, you can see the first two. We have this string, and then we can do njunc, and we'll get the number of junctions. And also index and subset the same way as we did G ranges. So if we want to keep reads which just map to the positive strand, we can do strand, G ranges equals positive, and we can use that to subset our GA alignments. And as you can see, we just get reads on the positive strand. We can also alter range positions for our GA alignments. And this is going to become really useful for when we do attack seek. If you want to manipulate your reads and say, I just want to take the first base pair of my read where it initially aligned to, we can use the narrow function, just like we did for G ranges. We just pass it our GA alignments function. Here we're going to say we want the start to be 1, and we want the whole thing to be a width of 1. And we can turn our reads now not into the full length, including the actual mapping, but full mapping of the read, we just get the first base pair. So now this is width 1, Q width 1. And what's kind of cool is it also has updated the cigar and the end junk for us. So now we had slightly complex cigars before, I believe. Yeah, like this. But now, because we're just looking at the first base pair, it's just one match. I guess we can also actually control this ourselves just by filtering down to the read first and then doing narrow. I don't think that's necessary because narrow actually takes into account the strand when it considers the start. So I think this is something Matt added. It's probably not that useful. If we decide that we're just sick of it being a G ranges alignment, and there will become reasons why that might be clear later on, especially with paired end data, we can actually turn our GA alignments object to a G ranges so that it really is a pure G ranges and we can act on it like a G ranges using the G ranges function. So we just give it our reads, we can tell it use mcols equals true, and then we just get a G ranges now. We don't have any junction information, but we have the range updated based on the full cigar string. So we know that this read was on this chromosome 12, the start of the read was here, the end of the read was here. We don't know anything about the mapping anymore. Did it skip? Did it have junctions? We know the string. RNA-seq reads, we don't want to do that, right? Because we know we have, maybe we're not worried about deletions, insertions, but we want to capture the information associated to junctions. So we can do that actually with the grg list function. Okay, and this is because a read potentially spans multiple exons. When we convert a read which spans multiple locations on the genome, we're going to have to create a list of G ranges for every read. And that's what this grg list function does. We just give it our reads, and it will return back a G ranges list. And every G ranges list there, or every element, contains the alignment for a read. So in this case, I'm just doing this for those which have an end junk of 1. So we know there's at least one junction in that read. So they should have 2 ranges they cover. The read is split into 2 because it has one junction. And we can see here for the first read, we have a G ranges covering what I guess is just before the splice site. And then we have our second ranges just after the splice. So we don't need to worry about just turning our split non-contiguous splice-aware aligned reads into G ranges. We can actually turn them into a G ranges list. We can also turn our G ranges lists or G ranges back into a genomic alignments object by using this as my G ranges object I want to convert and GA alignments. Okay. For whatever reason, if you're using things like Novo Align, there are software out there which just haven't adopted the SAM file format. You may have to read this yourself, turn it into a G ranges, and then turn it into a GA alignments by using this method. So now we have our full GA alignments, and we got things like cigar string and end junk, which just wasn't available previously. So effectively, this is the important thing, and we're going to do this for a taxi. This just means that you can convert from your GA alignments to a G ranges, perform some operations which you couldn't do on GA alignments, and then pass it back into a GA alignments object. So here we'll just do an example of this, and this is quite common. This is the concept of footprinting for a taxi. Take our reads, we turn them into a G ranges. I can then use our resize function, and that's strand aware. Take my G ranges, just fix it on the start position and set that to be width equals. Okay, so that's just going to take my very beginning of the read in a strand specific manner. And then I can turn that into back into a GA alignments by doing as, give it our GA alignments object, just sorry, tell it if there's going to be a GA alignments object, make it my G ranges. Again, this is a very specific example for a taxi, but there may be reasons why you want to be able to manipulate your reads prior to starting to operate on. Let's see what we got left. Working with large bound. That's great. So one really important reason why we want them as a GA alignments object is because if they're a GA alignments object, we can then export that back to a bound file. So G ranges we're only ever going to be able to export to a bed. But once we've manipulated this, maybe got it down to its first base pair, we can turn that back into our GA alignments, and we can then use finally the R track layer package, which is our IO package for Bioconductor or biological data. And we can export our GA alignments object. We just give it the connection, and it will write out directly to a bound file. A really nice feature about exporting through our track layer is then when it writes it out, it is by default coordinate sorted. So we don't need to sort it again. And it's going to produce the index while it creates it. So it's actually kind of a nice trick that Bioconductor builds those, and we don't need to make them ourselves. So again, like I said at the beginning, a lot of the time you're not really going to need to manipulate bound files. You're going to pass them from one piece of software to another, and that will handle much of the manipulation or the summarization of that bound file. And that's what we will do in our second session. But if you're ever going to do something unique or outside of this software, and I think a lot of you probably will do, you want to be able to operate on your big bound files, and then you want to be able to write them back. This, just like we had the problem with FastQ, we wanted to stream parts as we, rather than read the whole data into memory. Now we're going to have the same issue here, but luckily we can handle bound files, or we have some degree of control of bound files, of what we read from a bound files by taking advantage of that index. So we can use this function, which is a hard function to say, scan bound param, to really specify both what and where, I guess, which regions we want to import from our data. So the what information do we want to import, and the which, where do we want to import it from. So, we looked at these bound files earlier on. So this is a complete bound file, right? It's got all this information. Actually, a bound file is still valid if it has no sequence information or no quality for the sequence information. It's a pain, but you can still have a valid bound file now. The same way, that people do that to save memory. Why would I store all the sequence information if I have the FastQ? Maybe there are ways they know what they align them to, and they can just look up the sequence which was underneath the read later on. So we can control what information we actually import in from these reads. Previously, we haven't had any sequence information. When I read GA alignments, you didn't see any sequence information. You didn't see any read IDs, and you didn't see any flags. And that's because default, it just wants to read in this information as simply as it can. It just wants the ranges, really. What's really going to save us a lot of time and space is the which, because we can just select which regions we want to read, get our reads from. Okay. So to specify the which parameter, where we want to get reads from, it's going to accept a G ranges. Right? So we're going to give it a G ranges, and it's going to say, I will import reads from my BAM file, which are within this G ranges region. So we know how to build G ranges. We can just do G ranges, chromosome 12. And here I'm going to tell it, I just want to import over this region. And then I can create our scan BAM param object. I really love saying that word, scan BAM param. Sounds great. Scan BAM param object by just providing it with the which. We tell it we want to only read in from these ranges. So this builds our scan BAM param class object. And we can see it's got some information here. It tells us the class, tells us I'm not specifying any flags, no filters on really anything other than the which, where I just have one range. Okay? I can import multiple ranges if I want to, but here I've just set one range. So now when I go read in this file, I can do read GA alignments. I can specify the file, but I just specify param and I pass it the scan BAM param object I created on the last slide, so the one specifying the which. And now when it imports it, it doesn't read the whole file, it just goes straight to that region and reads it. Okay, super quick and super memory efficient. We can also control the what. Okay, so when I said earlier on, when we were looking earlier on at our GA alignments, we don't have any metadata columns here, right? We just got the information on the reads itself. So here now, if I specify scan BAM param a what, and I tell it I want the Q name, the seek, or the quality, when I then provide that scan BAM param object to my read GA alignments, I'm going to get additional information. And again, like a G ranges, we have GA alignment with one alignment and 3 metadata columns. And these are my metadata columns now. Q name, which is the character. So this is our read names. Seek. Okay, so we've already come across DNA string sets, and all the sequence information is now held inside the metadata columns. And we have qual. So we have the quality as a thread quality score, which is actually like a subset of the DNA string set contained here. Okay. So by default, we don't actually read in much information from the BAM. We have to explicitly state what we want to read. Okay. And as you see here, we can access that information the same way we would GRange's metadata columns using this mcols. A little bit of time. I just want to quickly show you. I can't quite remember where it is, but I do remember it's a pain to find out what you can actually read into a BAM file. So what are you able to specify here to read in? So let's actually just quickly go find the help for that. I think if I do the help for scan BAM param, it's actually going to point me to another page, but we can have a look. Okay, so scan BAM param, various information on how it does it. And if I go what character vector returning the name, naming the fields to return scan BAM what returns a vector of available fields. Ooh. Okay, so I can look at the fields described in scan BAM. Hopefully somewhere here there is a little bit more information on this. So here we have things we can actually get: Q name, flag, read name, strand, position, map Q, which is mapping quality. This is often very useful. You might want to filter out low mapping qualities. I size we're going to get later. This is your insert size as predicted. So we can get all this information. So you can choose what you want to import into your BAM file. It did suggest that by typing this in, it would also tell me what's available. Oh, there you go. So that also scan BAM what. If I use that function, it also tells me all the things I can specify to that scan BAM what arguments, right? So again, really useful. iSize we're definitely going to get. MapQ we'll use for filtering later. Cool. Group ID often relates to like a sample ID. Yes. So we can really import much more than we do, but by default, we're not going to get much. It's just going to be these basics of the alignment, but we can control that with scan bound param, specifying additional ones. Okay, so I think the last slide here is just an example then how we may operate on a full size bound file. Okay. The way we can do that is by taking advantage of the header. Okay, so the header tells us every single chromosome and its length. So let's just remind ourselves of that very quickly. So we got the BAM, I scan the header, and then I'll extract the targets from that header. If I look here, I only have one chromosome, but it's chromosome 12, and it tells me the length of it. So then in conjunction with that, I can actually build a range which would cover the whole of one chromosome. Right, so in this example here, what I'm going to do is loop through all the chromosomes in the header for I and 1 to length chromosomes. I will build little G ranges of that chromosome. So it will be the chromosome name. It will start at 1 and it will go to the length of the chromosome. And I can use that then as a scan bound param filter. I just provide it to which. And then when I read GA alignments, I'm not going to read in the whole file. I will just read in one chromosome at a time. Let me just show you what that G ranges to import is. So if I set I to be 1, oh, sorry. First time it's gone through the loop here, the G ranges to import is chromosome 12 from the very beginning to the very end of it. Right, so it's going to go in, read that to my reads, and here I'm not storing it as anything, right? I'm just giving the length. But you can imagine you could write out BAM files over time, operate or do a little bit more operations on these GA alignments. Just means you don't have to operate with the whole file in memory, just loop through one chromosome at a time based on the information we actually got from the BAM file itself. Okay, so I think that's the end of our session now. We have some exercises. I imagine they've got complex at this point. So please feel free to run through that. I will probably run through this at the very end of the day now, because we've got a bit of a longer session in the next one, I think. So that's an intro to alignment, right? And I'm really showing you the basics there. It gets deep. Like they've got some really great tools there, including what I haven't shown you is how you can really play around with the junctions. Right? So they've got very clever ways of extracting junction and read information. But we don't need that to do what we're going to be doing in the future sessions. So if you want to know, just ask me and I can always talk you through it. So we have our next session is at 2:30. I'm not going to run through the exercises before, so it just takes a little bit longer now. I will run through them in our final thing, which will be around 4:30. I will run through the exercises. After the break, we are going to come back and do summarizing. So this is how we count reads over genes, how we make bigwigs from BAM files. Okay. Then we're into RNA-seq chips. All right, so I'm going to pause my share. Stop my share. Hi, everyone. I will pause my record. Okay, so I'm now going to run through our last session in Bioconductor. So we finally got through everything we need to know to do the rest of our analysis. Importantly, in our last few sessions, what we have covered is really the raw data from FASTQ, how we can align this either as spliced or unspliced alignment, so really DNA or RNA-seq-like alignment. And then in our last session, we saw how we can manipulate these a little bit more memory-friendly ways, such as sorting using very low memory or very high memory if you've got a big computer. and also how you can really dig down into the band file and have a look at what's happening underneath. You know, look at the reads directly. As I said in my last session, though, a lot of the time you're just going to pass these band files you've created over to another function or an external program. So we don't really often need to look low level at the reads, but you may do if you're doing something custom or if you're doing quality control. So in this last session, we can take everything we've done, creating band files, looking at like coverage, RLE lists, view ranges, and put them all together to try and summarize signal from our band file over particular genomic regions. Okay. So as I just said, we've seen the data in FASTQ format. We have a read name, sequence, and then thread score. And that's everything we need really to go do an alignment. And once we've aligned our data, we have our records as SAM file, the binary being a BAM file. But if we look at the actual records, not the header we've already looked at in our last session, importantly, this contains all the information from the read, but chromosome, start, and then how that read is mapped. And so we know, really, given we've sequenced RNA-seq, we know where all the RNA-seq reads have mapped to in the genome. If we're mapping a transcription factor binding site, we'll have reads map into areas of the genome where our transcription factor may be found. So the next thing we really want to do, or a common analysis step in the interpretation of most high-throughput sequence data, is to try and quantify that abundance of sequence, either relative to other samples or relative to itself over in genomic locations. So as an example here, we have reads mapping, but we're looking at the coverage. We saw this in IGV, and we can see we have lots of signal over our exons. In RNA-seq, we're going to use our BAMs to help us assess the abundance of transcripts genes between differing conditions. And a very simple example here, we have two bigwigs we've generated. And you can see you have way more signal over this first gene in our HeLa cells, but a lot more signal in the second gene over our GM12878 cell line. Okay, so we might want to somehow quantify that difference, maybe get fold changes. Similarly, ChIP-seq, ATAC-seq, we're going to look for pile-up of reads. So when we have lots of reads in a region, for ChIP-seq, we'll often have a control sample, an IP input, which is effectively everything we do to these samples minus the antibody capture, and this helps us with things like background. And we may compare this region and these samples versus our input to say we have a binding site. For ATAC-seq, we may just compare these regions versus the local area, because these look peaky, right, versus the surrounding area, to tell us we have a binding site or maybe a region of open chromatin here. So far, we have used, we have a few different ways of really reviewing our data, and hopefully you've seen that one of the best ways we can quickly review signal over a genome is using a bigwig. Right now, we haven't produced a bigwig, I think. I don't think so. But we have imported them and we've got them from ENCODE and we've learned how to manipulate them a bit. And we know that we can look in IGV. In this case, we're probably just seeing signal over the beginning of genes, right? But once we've got it in IGV, it's super easy just to move back and forward and get an idea of how the data looks. And then we're going to use BAM files often to summarize our data as well. And I think it's just falling off the bottom of my slide, but I just mean we want to take all this information in reads here. This is our input control. This is our actual test sample. We want to take all that information and really lose it just to say we have an enrichment region here. And this is like a bed file at the bottom here. This region is enriched here versus here. Lost a lot of information, but this is the most important thing. We have a binding site here. So there are two main functions which will allow us to summarize our aligned data over the genome and within regions. These are the coverage function, which is really fantastic, and the summarize overlaps function, which is also really good. These are both actually, well, coverage is in lots of different packages. We will take advantage of the coverage function inside genomic alignments. This is kind of a bioconductor, I guess, methodology standard, is that you'll often have the same named function operating on very similar objects. We can use coverage on G ranges, and we can use coverage on our GA alignments, or we can use coverage directly on a BAM file, which will then create our base pair signal alignments across the genome. So effectively our bigwig. Summarize overlaps we can use to actually count the number of reads mapping to signals, or sorry, from signals or alignments within a genomic feature. So that's usually things like exons to genes, but it can also be reads over repeats. Okay, so in this session, we'll work with aligned data as BAM files. I provided BAM files we saw in our last session. We're going to use them again, and we'll use the liver body map and the heart body map. And these are the examples we have in IGV. So I've extracted them from IGV, realigned them using our methodology, and that's what we're going to see today. To make use of anything in the genomic alignments package, we know first off, we just need to, just checking I'm recording. We need to know, we need to load the library. So we've already installed genomic alignments to look at BAM files last session. So here we can just do library genomic alignments. And we should have then all the functionality available to us. The coverage function, as I said, is available in lots of different packages in Bioconductor, and we can use it with G ranges, we can use it with GA alignments, but actually it will also accept a BAM file path and will return an RLE list. Okay, so we've already seen RLE lists. These are ways of encoding signal across a chromosome, or RLE is a way of doing a signal across a chromosome. A list then will represent, we'll have a list element for every chromosome in our genome without. If we're going to use this coverage function, we need to do what we do with every different type of data or BAM file. We need to check whether it's sorted, and then we need to index it. Okay, sorting is going to allow us to indexing it, and indexing allows us to run random access. So in this case, I'm going to take our data in the body map directory, and I'm going to just create a file called heart.bam. We don't need to put the extension here. And then I will index that BAM file using the index BAM function. And this is going to return here the actual path to the sorted BAM. And then I pass that to index BAM. And we get the name of the binary index. And we actually have it's actually named. This is a named vector. This is the value of the binary index. And the name of that index in our vector is the name of the file we created the index from. Given we have now everything we need to start to use this BAM file, it's sorted and indexed, it's actually super easy just to create a RLE list. We just pass directly to the coverage function our sorted index BAM file, and it will create our favorite object, this RLE list, from the iRanges package. If we look at this now, we've already seen RLE lists. We covered this in the genomic scores package, but we can just remind ourselves what it actually has. We only have one chromosome in this BAM file. Remember, we just have chromosome 12. That's where this SLCA gene was. So we only have an RLE list of length 1. We only have one chromosome. But inside that list, we have, this is the name of the element, and we have our integer RLE. And just to remind ourselves, we have our lengths here and our values here. And this tells us that we have, for the first 33,666 bases, we have a score of 0. Then we have a little island of 50 bases where we have a score of 2. So we probably have 2 reads mapping there for 50. And then we have this long with 0, and then we have stretch of 35 where we have 3 reads, and then it goes on and on until we get to the end of the chromosome. This is a very efficient way of storing numbers, long stretches of numbers, and allows us to hold the memory kind of low when we're working with these. Very simple to create. You know, previously we've had to work from G ranges. Now we can just give it the bound file. So from that, we should have seen this already, and you could remind yourself by going back to our genomic scores. I'm not sure if that links there. We can remind ourselves by going back to our genomic scores lectures, that once we have this RLE list, there are some nice easy tricks we can do with it. I thought one which is really cool is we can just extract the RLE values itself. So we can get to this part of it out of the list by using our standard list accessor double square brackets, give it chromosome 12, and that will then pull out the RLE. We can use our standard indexing on RLEs if we remember. So here I can take our RLE vector and I can just pull out the region I want to look at. This is, I know up front is the region over the gene. And then if I want to do some plots with this, you know, we're going to show you how you can do this using ggvis later on. But we don't need ggvis to do this. We can just make our data frame, give it the same region we've extracted signal from as the position column, and then give the actual signal the result from extracting this region from our RLE. Okay, I'm going to times that by 1,000 so it's not so small. And then we pass this. And I think we have an exercise which looks very much like this, just working from coverage or load ggplot, and then we can pass it our new data frame, and we'll tell it plot on the X, the position, the Y, the signal, make us a geom line, make it theme minimal. And we've got our IGV style plot of coverage calculated from our bound file. Okay, so we're really doing some proper analysis at this point. The coverage actually also accepts, as I said, it accepts multiple inputs. It also accepts the GA alignment object itself. So if you may want to do a little bit of manipulation post-BAM, and a really obvious thing you might want to do is just extract the reads on the positive strand versus the negative, because then you can get strand-specific expression. So here, rather than just do it directly from the BAM file, we'll first read it in, we'll get our GA alignments object, and then that can be passed to coverage, and then we'll get exactly the same result. It doesn't matter if we go from straight coverage from the BAM file or coverage from the GA alignments we read in, it will give us the same result. Just the advantage is you have this intermediate object you can still manipulate. And we can also do this, as I was saying, for coverage. We can also use G ranges. And G ranges are very easy to build, so it's often nice to be able to use coverage from them. But we can also use G ranges lists. And the important thing here is that G ranges lists, as we saw when we were looking at aligned data in the previous session, we can turn our junction reads into G ranges lists. And that just means when we make coverage, we're going to be able to have coverage across exons, but we won't get any coverage for the junction itself. So this coverage function is super flexible. It takes G ranges lists, BAM files, GA alignments, and probably other objects too. Like I was saying, yeah. This means that we can do some operations on our sequences prior to creating our bigwig. And one, which is, I think, a nice recipe for you, is to split it into stranded versus non-stranded. So here we can just use our read-in GA alignments object from our BAM file. We can just filter it down to those which have a strand which equals plus, so on the positive strand. Once I've extracted them, I can run the coverage. And then I can here, I think I just want to show you it, so it's available here. But then I can use export.bw, which is our export for a bigwig. And I can pass it our coverage RLE list, and then just set it to write it to this file. Really usefully, the coverage function allows us in some way to add an additional parameter, weight. And now you can use that weight to down or up weight our samples. Okay, so we can use weight as a normalization factor. If we have more reads in one sample than we do in another, when we get the coverage, we're going to have higher coverage and sample with more reads, right? So if we want to normalize them and we want to compare samples side by side, we want to account for the fact one sample may have more reads than the other. So we can add weight to the coverage function by just adding this weight parameter. And here, if I put this to be 10, we can see that every time we have some coverage, so say here 50, it's 2. Here in the one where we've added the weight, we now have 20. So it's timesing whatever the coverage was by 10, and just allowing us in some way to correct this coverage or alter the actual coverage score. It's nice as it's doing it while it's actually generating it, right? So we don't need to worry about doing this post hoc. But we did see we can actually, times RLE lists by values if we wanted to. Now, using this in combination with what we learned last session actually allows us to very quickly generate normalized bigwigs. Okay, so how am I doing for time before I go into this? So, yeah, thanks. So once you've sorted and indexed your file, we know that the binary file, which is very small, contains the information on things like total number of reads. Okay, so we can use IDX StatsBAM in contrast to quick flag summary, which is really slow. This isn't going to read your BAM file, even though we're pointing to it. It's going to read the BAI file, the binary file, and it will just provide you with some very useful data frame of values, what chromosomes you have, how long they are, and importantly, how many mapped reads you have. Okay, so this is going to be our normalization parameter. We also have unmapped, which is useful here as well. So what we can do then is some way apply a weight to our data. Usually here we've only got one chromosome, right? Usually we would have a ton of chromosomes. We've only got chromosome 12 here just because it's example data. But what we would do is extract the mapped column from that data frame. So this column, get the sum of all the values, so the total mapped reads. And now we just need to apply that as a sensible weight. So we will do coverage, we will give it the BAM file, and then we will just do 1 over the total number of maps. In fact, here it's 1,000,000 over the total number of maps. So this is very equivalent to your reads per kilobase per million. It's just reads depth per million reads at this point. So then if we look now at our coverage, we can see we've got fractional values. But it means now if I've sequenced one sample way deeper than other, I can now actually compare them when I look at them in IGV, or if I use that bigwig in further summarization efforts. So it's this then that we could pass to, I'm not going to do it, but we could pass that normalized coverage now to our, what is our export BW function? To our export BW function, and then we've got a bigwig which is normalized in balance. So actually, if you're working in things like bacteria or prokaryotics, you don't need to worry so much about complexities in summarizing scores. You can often work directly from bigwigs. We have lots of labs here who work on prokaryotic genomes. So we've already seen that we can do things like summary statistics on our RLE lists. Again, it's in our genomic scores lecture. But one thing we might want to do here is just say summarize the score over genes or over exons. So we can do this very easily now using some of the tools we've already learned. So first off, we need to get some exons we want to count over. We know we have the TXDB package. This contains all the genomic information for our particular genome. Here, HD38, H. sapiens, UCSC. So I'm going to extract all the exons for the genes, right, as a GRanges list by giving it the TXDB of interest and telling it I want my exons by gene. Let's run this in the actual terminal maybe. Sorry, in the RStudio. 3 seconds to load the library. It takes a second to do exons. Okay, so then I should get exons of genes. I should have, I don't know, around 30,000 gene entries or something, 31,000 entries, because I have 31,000 genes within this particular genome. And if I was just to look at the first one, exons of genes, you can see actually I have a GRanges list now of just length 1. And the first element is our entrees ID 1. And these are all the exons which make up that particular entrees ID. Okay. If I want to subset then down to that entrees ID, I can do that by name. I can just do exons of genes, and I will do double square brackets, which means it's going to extract the actual value rather than the list. And I'll give it the name of the gene I want to extract, which is SRC25A3. Note, I'm not giving a number here. I'm giving a quoted string of a number. If I put the number there, it will take out the 500 or 5,250th index, right? So I need to actually put it in quotes to pull out the name. And it'll be the actual entrees ID. So with that, then, I can use these tricks we'd already learned in our RLE list genomic scores lecture. I can extract the coverage as an RLE list directly to my heart coverage norm. Can I try that? Do I actually have that object available to me? I don't think I've made sort data. Sorted. I don't know. Sorry, I'm just going to quickly see if I can do this. I don't have the heart. So I don't have the data available. I'll do it in the break. But effectively, this will create our RLE list object of coverage across the heart band, which is our sorted band file. And then we can take the mean, and we can apply our RLE list operations. But first, we can subset our RLE list down to all the exons across that gene. So we can give it these GRanges, and it's going to extract an RLE list from here, and we'll have an entry for every exon. And then we can get the mean, which will give us the mean signal across all the exons for that gene. Okay, so already we can get an idea of just the signal over exons for that particular gene. And then we can start to compare that across samples. Well, really though, if we want to compare across 2 samples, probably the best way, or multiple samples, the best way to do this is summarize it. And I say it's the best way because it's so well integrated with Bioconductor tools. The output from summarize overlap feeds directly into DSeq, EdgeR, and other Bioconductor tools, and it takes all these objects we really like. There is an equivalent, and let me know on subread if you're interested in learning this. I don't think we cover it until we get to the RNA-seq. But our subread, which is our aligner, is actually most famous for having the counter inside it, which is called feature counts. Let me see if I can find that here. It's feature counts. So subread, a lot of people don't use it for alignment. They just use it for counting. And we'll cover that a little bit later on. But summarize overlaps is just so much more friendly to use inside a Bioconductor setting. Probably a little bit slower, but we can parallelize it, and it will give us effectively the same results. Feature counts compares themselves to summarize overlaps to make sure it makes no difference. Important thing about summarize overlaps, which we haven't really learned how to deal with yet, and it's going to take account of all this for us, is it will handle reads overlapping multiple regions. So if we have one read which spans and goes to 2 exons, we don't want to count it twice, right? We only count those 2 parts as one read and we assign that to the full gene. Another thing is this idea of assigning reads to meta features. So we don't necessarily want to line reads to exons again, but we want to summarize all the exons over up to a gene. So we have a gene score. Let me just open IGV to show you maybe why that's a problem. I can show you why it's a problem here. So just to show you a little bit of these examples, here we have reads which map from one exon to another, but we also have some reads which maybe go into the intron. Right? These are probably not great reads. Maybe they're just spurious alignments. We want to be able to count these reads spanning exons and include them in our overall count, maybe discount some of these ones in introns. We also need to be able, given a gene model, if we look at our gene model here, we can see we have exons which are in multiple transcripts, and they kind of overlap each other. And sometimes they will be like here, you will have some overlapping portions and a portion which is unique. We don't really want to care about that. We just want to know how many reads align to any of these transcripts, any of these exons, and summarize them out to each other. So we need a tool set to do that for us. And the one to do it for us is this tool, Summarize Overlaps. When using this tool, by default, and the way we'll run it, a read can only be assigned to one feature. So meta feature would be the, meta feature here. So whereas we would have reads aligning to this read would align to multiple exons, we would only want to count that once. Sometimes we'll have reads which span multiple genes. We want to throw those away because we can't be sure which read or which gene that read is truly allocated. So summarize overlaps is just going to allow us to count a read which can be assigned to one feature. This is really important when we start to think about overlapping exons, but we'll see this in much more detail when we actually get to our RNAs. Like I was saying, summarize overlap is very well integrated. I think I spelled integrated wrong. Integrated with many of the bioconductor objects we can see. So as with coverage, it can count reads from BAM files or GRanges objects. So if you want to do something normal like work from a BAM file, that's easy here. If you want to some way maybe count bed files or peaks in regions, you can still use some of those summarize overlaps. You just import it as a GRanges. If you decide to do something weird to your BAM file and you want to turn it into a GRanges, manipulate it, you can still use that GRanges object. Summarize overlaps then can take a BAM file or a GRanges to count from, but counting over, it will take either a GRanges, so features such as exons, or GRanges lists. Right? A GRanges list, we just had one. This is our GRanges list. And rather than summarizing over the individual exons, it will summarize the signal over all these exons up to the gene. So summarize overlap effectively allows us these 2 wide modes, one which is features, and one which is meta features. To count non-overlapping regions, we can simply specify the path to our BAM file and the GRanges we want to actually count over. And then this is going to give us back a new object called a ranged summarized experiment. And summarized experiment objects are the basis for differential RNA-seq, and the differential really attack-seq as well. And actually the framework for single cell objects, single cell summarized experiment objects. Okay. The really nice thing is that by using the summarize overlap function, we don't have to recreate our range summarized experiment object. It is going to do it for us. So first we need something to count over. Here I'm just going to count over promoters. So I will extract all the genes for our TXDB H sapiens UCSC H238. Let's This will give us all the genes, and it gives us the full length of the gene body. It doesn't know about exons when we do it this way. We can then pass that output to the promoters function, and promoters will take into account the strand, positive, negative, and then it will go downstream or upstream a certain region around the TSS, and then that will give us what I'm defining here as our TSS region. If I then want to count over those regions from a BAM file, so I want to know how many reads land in those regions, I can just pass this G ranges to the summarize overlaps function as the first argument, and the second argument I can give it the heart.bam. Now I will have my first ranged summarized experiment object from the summarized experiment package. And this range summarized experiment object actually contains some really useful information. It contains both a matrix of our counts from our BAM file over these regions of interest, and because it's a ranged summarized experiment object, it also contains the original G ranges we did the counting over. As with other bioconductor objects, if we just type this in, my TSS counts, we get, a sensible description. It tells us we have class, range, summarized experiment. It tells us the dimensions. And in this case, we have 27,000 rows, one column. So we only have one BAM file we've counted from, but we have 27,000 promoters to look at. We have no metadata. That will come when we start to do RNA-seq analysis, attack-seq. We will add metadata, which will be information like groups, what we want to compare. We have one assay, which is our counts. So that's what we've just created. And then we have row names. This is our names for the matrix, and they're in the order here, just alphanumerical. But this is the entrees D gene IDs. Even though they look like numbers, that's alpha globulin ID. I don't know what 10 is. We have row data, which is a little different from row names. And this is your G ranges information. And then we have col names, which is the name of the sample. And then we would have col data. Oh, sorry, I'm wrong. So metadata actually just stores random information, metadata information about this. It's actually column data, which stores the information on your columns. So col data, we may say that the heart BAM is part of a heart group. So that's where we would do define our groups for differential expression analysis. And one of the most useful things we want to get out of this is that matrix, okay? Because we may not be doing our analysis in R, we just want to get hold of that matrix. And the counts we see here are stored in our assays element of our range summarized experiment. And then in order to get the matrix back out of this, and remember this is a matrix, not a data frame, we can use the assay accessor, and we just give it our range summarized experiment object, and this will return a standard matrix. And if I just look at our gene of interest, our 5,250, we can see we just get that row back, and it's named by the row names and the name of the gene. The column is the name of the sample, and we have 2,934 gene reads which map to the TSS of that gene. You can also extract the row ranges for this. Okay, and this isn't so useful necessarily for RNA-seq, but it's really useful for attack-seq. Just by using the row ranges function on our summarized experiment object, and it's going to give us back in the same order as the actual matrix all the ranges that we counted over for doing this analysis. Gene models, though, we actually want to do this summarization to a meta feature. We're not too interested in just summarizing over exons often, unless we're doing differential exon analysis. That's a little bit complex. We'll show you that later. In this case, we want to summarize all the reads for a whole gene, but we don't want to count reads which overlap similar exons. So if a read maps to 2 exons which are subtly different, we don't want to double count them. We want to just count them once for the whole gene. So we can use the fact that summarize overlaps accepts G-ranges lists to do this. But first, we need something to count over. So what we'll do is we'll take our TXDB object for H. sapiens UCSC HD38, and we'll extract again by gene our G-ranges lists of every gene and all the exons in it. If we just look at gene exon for our SLC25A3 gene, we can see that we have here the name of our entrees ID for this particular entry. It is a G-ranges list, and we have all the exons for that gene listed there. And you can see these are overlapping. The first one, for instance, it's the same endpoint with different exon start points. This just represents different transcript variants of the same gene. To summarize that, we can just do exactly the same. The only difference now is that we have given it a G-ranges list rather than a G-ranges. Previously, we gave it this G-ranges my TSS counts, sorry, TSS. Now we're giving it this G-ranges list. We can just run summarize overlaps again. We don't need to give it any more parameters, and we don't need to change anything. We tell it to count from the BAM file, and I will get the same object back, a range summarized experiment with, again, 27,000 rows. Right? But I'm not counting over genes now, but gene TSSs. I'm summarizing the whole gene body, all the exons in that gene. No metadata. Same assays, though. I still have my counts in the assays, same gene IDs. We will have different row information, same column information. If I go now and I extract from this the assay, so the counts. So if I just do assay on my range summarized experiment object, it will return the first assay. We only have one assay. If we want to return multiple assays, we could use assays. But here we just do assay, and it gives us again back our count matrix, except now we're going to have more counts because we've counted over the entire gene. Same setup, same matrix. So this is exactly how you would do RNA-seq analysis. I'm going to show you this later, but you extract the exons by gene, and you just run this summarize overlaps over those G-ranges list using your BAM. Simple as that, and then you've got the input for downstream analysis, or you can extract the matrix from the actual object and export that, or do with that what you would. The row ranges now contain slightly different information, but it is the information we want. So if we look at just the first one here printed, you can see we've got a G-ranges list the same length as our rows, right? Except every element now in this row ranges we've returned is actually a G-ranges list representing all the exons we've counted over for every row. So this is our gene 1, it's our first row in this object. And these are all the exons we counted over. It's nice to have that in the same object because you then might want to pass that to something else to use. Just have all the information, the actual counts, the BAM file I used, and the ranges I counted over all in that one object. A nice feature of summarized overlaps is that we can actually just count across multiple BAM files at the same time. So I'm not sure if I'm going to cover it here, but I'm sure I will cover it later on. I will allude to it now. So we can count across multiple BAM files. All we need to do is specify a vector of the BAM files we want to count over. Okay. So then it will go and count over all these. I'm not going to cover it in today's session, but you can imagine this is something which is very easily parallelizable, right? Parallelizable, that's the word. But we can set two different jobs off, one counting over the regions in heart, one counting over the regions in liver, and then just aggregate them. So we'll look at a little bit more about parallelization in some of our later sessions. But what's really nice about this now is that by running them together, we get one matrix. And we have the names of the actual files we've counted over here. And for this particular row, this gene of interest, we have the total counts. Okay. We haven't done any normalization to total reads or anything, but already we have a way of comparing total reads over genes between these two samples. Okay, So like with everything, let's try and look at the low memory version of this, because in reality, if you want to do this over 100 files on your laptop, it's going to perhaps struggle. Or if you want a really high performance machine, maybe you can speed it up by setting your memory higher. So counting as from BAM files, as with our sorting, can be configured to trade memory use for speed, right? The more friendly you are with memory, so maybe the less memory you set, the slower it will be, higher it will do it super fast. We can actually control the amount of memory used by creating an object which kind of wraps around our BAM file. It's actually called a BAM file object. Okay. So to create a BAM file object, all we need to do is pass the path to the BAM file to this function BAM file from the RSAMTools package, and it creates a new object, a BAM file, as I just said, from the RSAMTools. The really cool thing about this is that for these objects, we can actually set how much of this BAM file is read in at a time. Okay, and we do that by just setting this yield size parameter here. And this controls, as I just said, how many reads are held in memory to do the counting. And then it will throw those out of memory, and then it will take another 1,000 here in this case, do the counting, another 1,000, do the counting. And it will just build this up rather than reading the whole BAM file into memory. So to control this, we just need to set BAM file, give it our BAM file, actual path to the BAM file, and then we just set yield size. And here I've set this deliberately low, it's 1,000. You can set this to however much memory you have on your computer. And then once I've created this BAM file object, I can just pass this into the place where I was putting the file path to the BAM file, just as if it was the direct, just as if it was the actual file path to the BAM. I can do summarize overlaps again over my gene exons, and it will do this probably slower here, but it will do it in very, very little memory. And the result will be identical. So very cool. Taken together, we can also supply BAM file list. So if we don't want to go through and do a BAM file for every single object we want to count from, we can actually provide BAM file list, and we can give that a vector of the BAM files we want to count from, and we set the yield size. And then when it goes to go do the counting from all of these BAM files, it's only going to read in 1,000 reads every time. This will then create our range summarized experiment object, and it's from there that we can start to do our downstream analysis, RNA-seq, PAC-seq, differentials, single cell. Okay, so that's actually the end of our session for today. I have some exercises to work through. We have now completed everything we would need to know to do RNA-seq, TAC-seq, ChIP-seq, and single cell. So from now on, we're really just going to be reusing these techniques, but in context. It's actually a really good way of learning, I find. Take this and then apply it. I would advise for everyone still here or listening, read through these notes again before we move on to RNA-seq, ChIP-seq, and TAC-seq. What I have deliberately done is put solutions to our future problems in the exercises as we go along. So I recommend just follow along the exercises and the answers. I'm obviously going to run through this probably around 4:30. I will see who's in the room, because sometimes I come to the Zoom to run through and there's no one there. Me talking to myself through the exercises can be a bit sad. But I've still put a video up of the solutions. I might just find my wife and make her listen to me. Monday, we will start the actual analysis, and we will start to work through some new sessions, and I believe we will start with RNA-seq. Okay, so I'll see everyone or I will see whoever's here at 4:30, and I will stop my sharing.